So CGRP is exciting because it is a potent vasodilator. CGRP has potential for patients in migraine. So the studies we've conducted, the two that are the background for my talk, one is a phase 1 study that was conducted in healthy volunteers. And the primary purpose of that study was safety and tolerability of the molecule. So we're looking to understand if galcanezumab could be safe and tolerable by healthy volunteers. So that was the first step. And then following that, I'll be talking about a phase 2b study. And in that study, we were looking at patients with migraine and understanding how their migraine efficacy with placebo versus galcanezumab correlated with reduction of migraine headache days. So galcanezumab has completed phase 2 trials, and there's going to be information on that in a poster this afternoon, more specifically. And we are currently in phase 3 trials at the moment. So the phase 2b migraine study for galcanezumab showed that there was a reduction in overall migraine headache days. And it looked at various doses from 5 milligrams up to 300 milligrams compared to placebo. And so at 300 milligrams and 120 milligrams versus placebo, there was a reduction in overall migraine headache days across a 12-week period of time. So the phase 2b study for galcanezumab included migraine patients that had between 4 and 14 headache days per month. And we studied a range of doses for galcanezumab to understand further safety, tolerability, and try to understand efficacy. We were looking for overall reduction of migraine headache days. So patients in the phase 2b trial were aged 18 to 65 and had headache migraine days between 4 and 14 per month. The galcanezumab study results showed that there was a significant reduction of migraine headache days per month over the 12-week period of time that it was studied. The doses studied of galcanezumab were 5, 50, 120, and 300, or placebo. And the doses were given once every 28 days for a period of 12 weeks. We're very fortunate that we have a biomarker that tells us something about CGRP target engagement. So we have a pharmacodynamic biomarker that tells us whether or not we've hit our target. And it teaches us mostly about dosing in that case. What we learned from this study that we're presenting today is comparing the actual biomarker study with the phase 2b efficacy study. Tells us a little bit about what happens when we take a healthy volunteer population and we use a biomarker to understand pharmacodynamic engagement and correlate it with a disease state. In this case, we're talking about correlation of healthy volunteers with migraine patients and trying to understand how CGRP behaves in these populations. The biomarker tells us we engage the target at certain doses, and when we looked at the phase 2b efficacy, what it suggests is that you need a real maximal effect of your biomarker. So you need to have really heavily engaged CGRP in order to expect it to play out in efficacy. Now this data is only in migraine patients at this point, so that's all we can draw a conclusion on is this one study in migraine patients and how it correlated. But it suggests that we need to be very cautious when we're using biomarkers for specific dose projection. When we're looking at doses with the biomarker, the capsaicin-induced dermal blood flow biomarker, where you don't have complete inhibition of the dermal blood flow effect from capsaicin, then we need to use caution in how we're going to interpret that to lead to dose efficacy projection for various indications. In this case for migraine, what we saw was that the higher doses where we saw complete inhibition of the capsaicin-induced dermal blood flow response did play out to be efficacious in the phase 2b trial with migraine patients. However, doses that were a lower effect, somewhere around ED90 or below, did not play out to have a significant effect on migraine patients, even though there was a numeric difference compared to placebo. So the take-home message of this is biomarkers can be very useful to understand disease, and in this case, to be able to understand something about a medication and how it behaves. We're still learning how biomarkers can correlate with disease states. And in this case, being a pharmacodynamic biomarker, we learned a lot about how the drug behaves that then helped us understand what we could use in our trials with patients to better indicate what drug doses we should use, but maybe more importantly, which drug doses we would expect may not have clear efficacy endpoints.